Yeah, so thank you, and it's great being here and present some of the data we have accumulated over the years. So I've been interested in sort of basic mechanism of muscle fatigue and work with that for almost 30 years now. First doing frog experiments, which was the animal of choice when I started, and then we turned to mouse, and I've been working with mouse muscles for 25 years or so. And I'm going to tell you a bit about what we've learned during these years. Try to skip the sort of fine details and go for a more global picture. But then, of course, this is sort of a global picture in my mind, having worked with this for 30 years or so. So please ask me and interrupt if there are things that this is just impossible to understand and so on. But I try my best to try to get the big message across. So I worked with studying muscle, isolated muscle and muscle cells for quite some time. In recent years, I've also started to study muscle fatigue in myself. So I've started with long distance running at the age of 50, stuff like that. So this is from a marathon some years ago where I managed to come 100 meters behind the number 3, so I missed the medals. But on the other hand, this was my personal best on the marathon at the age of 54, which not that many did their personal best then. They did it a few years when they were a bit younger. So I still have the future ahead of me in this respect. So now, if we muscle fatigue and just start to think about it in general terms. So here we have two different curves. You have a force power on the y-axis and time here. You can look at fatigue at different ways. The normal applied way of thinking of it is that you have a required force of power output, and then you can continue with that until you reach a state where you are exhausted. So think of this like a 400-meter run. For the first 300 meters or so, if you don't start at a ridiculously high speed, you continue rather happily, and then when you come to 322 meters or something like that, you reach this point where you can't keep up the speed, and then towards the end of the race, then some people seem to stand more or less still. And that is really when muscle fatigue has caught you, reached the point of exhaustion here, so you can't really go faster. The brain can convince the muscles to go a bit faster for a few steps, but for slightly longer you can't really do it. On the other hand, when you look at fatigue the way we do it, the upper curve here, maximum. So when we stimulate our muscle cells, we make them contract as much as they possibly can, then they relax for a while, and then another maximum contraction. And then, as you can see from here, then fatigue occurs directly. The second contraction, already then there is fatigue. But if you're out running or so, you don't note that because you don't need the maximum power all the time. But you would notice up here that, for instance, if you're weightlifting, if you do your personal best, you put it down, then you can't do it again because next time you try, you're weaker, meaning that you have some fatigue then. And here you needed the maximum effort. So if we then look at in training aspects and also in muscle diseases and so on, one can think of it, how could you do to increase your fatigue resistance? And based on this, that can be done in different ways. For instance, weight loss. If everything else is constant and you lose weight, then the required force power goes down, meaning that you can continue for a longer time until you reach the exhaustion point here, and you will then improve your performance. So you get more fatigue resistant, not really because your muscles are more fatigue resistant, but because you don't need to use them as much as before. So for someone with poor muscle function and so on, walking up the stairs could be something that's basically impossible because you reach this step here. You lose weight, and then you can do it. Strength training is another way to increase your fatigue resistance, maybe. Everything else constant, you with strength training, you will move this curve upwards, and then you can continue in some types of exercise for a longer period before you reach exhaustion. That, of course, is if, I mean, many times with strength training, if you get bigger muscles, you also increase in weight, and then you have the required coming up at the same time. And then the obvious one with endurance training, then you would sort of move this point over here. So the time it takes to come to this point will be delayed. So in that way one can think of it both in sports kind of environment and also in patients with muscle problems or other problems, that there could be different things. And in many patient situations the complaint is early fatigue, but most of the time I would say it is not really because this one comes too early. It's because muscle weakness in the first place. So you start with the muscle capacity down here, and then you reach exhaustion where you can't perform anymore very, very early because you were so weak to begin with. So you used sort of 60–70% of your maximum force power already in sort of common day things. But also in sports things, I think to think along these terms could also be useful in that maybe endurance training is not the only thing. If you're too weak, then you will have problems with the maximum instead, so to speak. So now comes some hardcore physiology here, that how to look at it when we come down to the mechanisms. So normal activation of muscles starts, of course, with the nervous system, central, number 1 and 2 here. And you can see that this is a cartoon made by a muscle physiologist. So the brain and all the motor systems and so on, that's nothing, just a number here. So here, of course, the brain decides that we should activate our muscles. Activate the motor neurons that send out their axons and activate the muscle, and here we have a muscle fiber. So fatigue, of course, could be in the nervous system and call that central fatigue. And it's always debated if fatigue really is a nervous kind of phenomenon or if it sits out in the muscles. And if you ask me, a muscle physiologist working with the muscle cells down here, of course I would say that the muscle part dominates. And if you're more interested in the neural aspects, you will have another opinion. But I think really most things in us and in biology, they are matched. So you don't have sort of enormous success out here and then you stop up at the central and vice versa. So I think they come together. I think it's very seldom that you have central fatigue without any problems whatsoever out in the muscles. But sometimes the central component can dominate a lot, like for instance, long distance endurance kind of sports in a hot environment. Then clearly the brain turns off the muscles prematurely because you're starting to heat up. If you cool down your head, you can perform better, meaning that you fool the brain that it's not that hot anymore, and then it can send out the signals to the muscles again. Whereas the muscles, of course, they don't care if you cool your head or not, so they face similar problem, but you can activate them more by cooling down. So then when we come out to the muscle, where I'm going to spend the rest of the time basically, what's happening out here? First you have the neuromuscular junction, where the nerve is activating the muscle fibers, and here you have an enormous safety margin. So there I would say that nothing happens. See, if you have an action potential which drives this thing coming down here, it will also come and spread to the muscle fiber. The only situation this doesn't happen is with the disease myasthenia gravis, where you have problems in this region. So then in the muscle you have action potentials traveling along the muscle fiber into the T-tubular system. In the T-tubular system it activates the sarcoplasmic reticulum to release calcium. The calcium activates the regulatory proteins, and then the motors, the cross bridges, start to work and the muscle contracts. And to stop this, you pump the calcium back into the sarcoplasmic reticulum. If the activation from above stops at the same time, calcium disappears and it goes back into the sarcoplasmic reticulum and the motors stop acting. Some of you have heard of this before, I imagine. And everyone has heard of this before. Not really. But this is the basic concept. So what's happening inside the muscle fibers? And what I will try to explain to you then, how you can look at mechanisms. And then the calcium will be a central component here. So when you have activation problems, then you don't have enough calcium to drive the motors down here. And then you can also have problems with the motors down here. So you have the calcium, but they don't still cannot perform maximally. So that's something we can distinguish when we come to the mechanisms. And the way we do our experiments is that we use single muscle fibers most of the time. And this is our favorite preparation. So it's the toe muscle. This one comes from a mouse. We also use from the rat. So you take out the toe muscle. And then you decide that I want, say, this fiber here. And then you dissect away everything else. So you only have one fiber left seen down here. So you do that with a forceps in one hand and a pair of scissors in the other one under a microscope. And it takes some time to learn this, but if you know how to do it, it's pretty quick. And then you have a perfectly happy contracting cell down here that you can stimulate with electrical pulses. And the cell down here then believes more or less that it's the nerve that activates it the normal way. So it contracts, and when it contracts, we can measure the force that it's producing. That's why we have to go through the palaver of dissecting it. If you can get thousands of cells with enzymes instead, but then they don't have any tendons. And if they don't have any tendons, you can't measure the force. So to measure the force, you need to dissect. One can have a motor here too, so you can do dynamic contractions when they're shortening and lengthening and so on if you want that. And we combine this with using a fluorescent indicator. So you can inject a fluorescent indicator into cell. Here's a microelectrode used for that. Here is a fluorescent cell. And with these fluorescent dyes, we can combine the force measurements with measuring calcium, which is something I would talk a lot about here. We can also measure other things, for instance, reactive oxygen species or free radicals, which are popular these days, and we've done quite a lot measuring them. And I will come back to that also quite a lot. So this is our basic method then, that we combine force measurements with something else and then try to figure out what's going on. And the first time we published this with using single mammalian fibers like this was actually 87. We came up with this method, and basically no one has repeated it. There have been a few labs that have done it over the years, but not regularly. So in that way, we're very fortunate. We're using a model that no one else uses. So whatever we do, we can say, oh, this is the right way, because no one else can check us, basically. But when they do, then most of the time, it turns out that it was okay in the first place. So this is then a way that a fatigue experiment can look like. So this is a single mouse fiber doing a 5 or whatever it could be, 6-minute, very intense exercise. So each spike here is a maximum contraction. So it contracts titanically. It can't contract any more than this. And it does it for 300 milliseconds. Then it waits for a while. And then it contracts again. And then it looks like this, that to begin with, force goes down rapidly by 10-15% or so. Then comes a rather flat portion here where it's sort of steady state. And then it goes into this exhaustion when force is dropping rather rapidly. And it always looks like this. It looked like this in the frog fibers, mouse fibers, rat fibers. We have done this in one small piece of a human muscle too. Couldn't get single fibers, and most humans want to keep their muscles, so it's not easy to do it on humans, but it looked identical in the human situation too, in the one experiment we've done. And when others have done similar things too, it seems to look like this. And if we now look at the calciums, so the calcium that's activating the contractions here, it starts, this is the first contraction. The calcium looks like this. Then number 2 here, calcium is actually bigger, meaning that the calcium is increasing while force is decreasing. So here we then can say that the problem to begin with is not the activation, it's downstream of the activation, it's the actually motors and so on that cannot produce maximum force anymore. Do you follow? So that is actually, so you can't do much by sort of motivation and so on. The early decline in force is due to something directly on the motors that you can't counteract if you do a maximum contraction just by putting in more effort, so to speak. But then when we come to exhaustion down here, then calcium has gone down and therefore also force goes down. So the activation fails at about this point. Here we put on caffeine. So if you put on caffeine, you can bring calcium back up again. And then force also comes back up again. So with coffee, you can actually counteract this activation problem in severe fatigue. You only need something like 1,000 cups of coffee here. And then you get the force back up again. The problem though being that this is sort of a safety mechanism. So you can get it up here for 3 contractions, but if you continue for a few more, it goes into contracture and dies. So this will, if you manage to drink 1,000 cups of coffee towards the end of your race, you have a few steps there where you really can perform, but that's the end of you. So the final spurt, one could say. So caffeine directly activates the calcium release mechanism, and you can bring it back up again. So showing that the fact that calcium is down here is not because it disappeared. It's still there, but it's not being released because you can get it back up again. So try to convince you that this makes some kind of sense then, that why on earth would anyone be interested in fatigue in single mouse toe muscle fibers? Does this have anything to do with fatigue in human muscles and so on? Well, the things we do here you can't do in human muscles, so that's the problem. What you can do in human muscles, and this is just one example, that you can stimulate electrically directly on muscles and then measure the force. And this has been done here in human muscles, in the quadriceps and the adductor pollicis, long time ago, but it's been done more recently too. It still looks the same. That with low stimulation frequencies you have low force, and when you increase the stimulation frequency, the force increases until you have everything being activated up here, and then it doesn't matter how much you increase the frequency. So this is something you can do in the intact human system with electrical stimulation. With the voluntary activation, then it becomes more complicated to study muscle function as such, because then the nervous system is pretty clever, and if the muscle, something happens in the muscle, the nervous system many times correct that, so you have another input to the muscle, and therefore more difficult to interpret. If we then do the same thing in our mouse fibers, we can stimulate them also at different frequencies, and we'll then get a rather identical kind of curve. But the advantage in our experiments is that then we can measure calcium. So instead of having frequency here, we can have the driving calcium concentration, because the relation between calcium and frequency is basically a straight line. So you can then go from having something that's just a number here, that's something that really matters, so to speak, the thing that drives the motors. And if you do then, so then you have force frequency curves, and under control conditions, by stimulating at different frequencies, we can get the control curve. And then when we do the way we did fatigue, the previous one, we start up here. And then as we stimulate, first calcium increased, whereas force went down, and then towards the end, calcium decreases and force goes down. So you will have another force calcium curve in the fatigued state. It's going down on the top there, meaning that the motors have become weaker. Doesn't matter how much calcium you have, the motors still cannot produce 100% force. They can only produce 80 to 90%. And that's nothing you can do anything about. That's just the fact that when you start to activate your muscles, the maximum force comes down. Then you also shift the curve towards the right, so you need more calcium. You need a nervous system, if that's still around, to activate the muscles more to get the same kind of force. And then when calcium comes down towards the end, you fall down rapidly here because the relationship is very steep. And you should keep in mind then that the nervous system is normally working on this region here, where it is very steep. So therefore this with central fatigue and so on comes into play, and it's very difficult to assess in that the nervous system is always on this part, and in fatigue also on this part. So if you put in a bit more nervous input, you increase because you can increase the calcium a bit. Whereas if you use the maximum, then you can't do much about it. So now to start to think about mechanisms here. And if we look at acute fatigue, the kind of fatigue you sense at the end of a 400-meter sprint or something like that, then that's very much related to anaerobic metabolism, meaning that you do your performance at the speed or intensity that's above what the mitochondria can cope with, above your aerobic capacity. And what will happen then is that you will have accumulation of lactic acid and breakdown of phosphocreatine. That will more or less come in parallel. And we now try to figure out which of these two is the important one. If we start with the lactic acid. So lactic acid doesn't really exist in muscle. You have lactate ions and you have hydrogen ions. And the lactate ions most likely does nothing except for being osmotically active and so on. Whereas the hydrogen ions, you have a decreased pH. That's supposed to be something that does something that might be dangerous, so to speak. We looked at that, and we then induced acidification by increasing the carbon dioxide in the solution where our cells are swimming. So with 5% carbon dioxide, that's the normal pH. We did fatigue will occur like this. We let the cell recover. We increased the carbon dioxide to 30%, meaning a pH decrease of 0.4 pH units, something like that, about as acid as you could be in severe fatigue. Fatigued it again, it became more fatigue resistant actually. Then back to 5% and something in between there, meaning that if you make the muscle cell acid, it performs as well as when it's not acid. So lactic acid doesn't really do anything to muscle performance. And we've done many other experiments along this pathway too, and also others have done, and it seems pretty clear that skeletal muscles, they're made to work when they're acid. They don't care much about it, and so there is something else that really causes the fatigue. And this something else then should be related to phosphocreatine, and the problem there is when you break it down, you have phosphate ions. And phosphate ions, they are the product in all energy-consuming reactions. And they really can do something. So one experiment to look at this is that we had muscle cells without creatine kinase, so they can't use their phosphocreatine. Normal fatigue in a wild type, a perfectly normal mouse. So force goes down, whereas calcium goes up to begin with. And then exhaustion, calcium down, force down. And in this case, it took 88 tetanus to reach to this state. And then we had cells without creatine kinase. And after 10 contractions, absolutely nothing. After 100, absolutely nothing. After 300, actually, something started to happen. So very much more fatigue resistant if they can't break down phosphocreatine. A bit on the slow side to begin with, but very, very fatigue resistant. So they sort of become a Paula Radcliffe kind of muscle cells. A bit slow, but can go forever. When you work with these kind of genetically manipulated muscles and mice and so on, there are all sorts of adaptations going on. So this could be that it has nothing to do with the lack of creatine kinase and breakdown of phosphocreatine. It's because some other kind of adaptation. So to check for that, we did this experiment. So we injected creatine kinase into muscle cells that don't have any creatine kinase. So we turn them from a knockout cell to a normal cell instantly. So they have no time at all to adapt and do something about it. And what we saw then is the red symbols here. So the normal ones, the white circles, calcium goes up and then comes down. The creatine kinase knockout, absolutely nothing happens during fatigue. When we inject creatine kinase into the creatine kinase knockout, calcium went up and then it came down. So it went towards the wild type. Look at force. Force goes down in the wild type. Nothing happens in the creatine kinase knockout. When they can break down creatine phosphate again, force goes down a bit. They're still more fatigue resistant, but that's because they have adapted and have more mitochondria. But they go towards the wild type. So if you block the creatine kinase, you can go for, if you're really into long distance endurance kind of things, it's great. If you want to do sprint exercise, on the other hand, then these guys, the creatine kinase knockouts, are absolutely useless. So they can't do the first sort of 50 steps in a sprint or so, they would be ways behind. But then the system gets going. So to conclude from this, that increased phosphate because of phosphocreatine breakdown is really an important cause of acute muscle fatigue. There are also other mechanisms involved, but I think this is, if one has to pick one, it's this one. Whereas lactic acid has no or little impact on skeletal muscle function. Marker, but not a maker. Something I've stolen from Bengt Saltin, who was here to give this seminar some weeks ago. So it's so easy to measure it. You can measure it in the blood because it comes out of the muscle cells. The phosphate ions, they don't come out of muscle cells. You need biopsies to find them and so on. So when you have lactic acid coming out, you use aerobic metabolism, you have problems inducing muscle fatigue, but it's not really the lactic acid that does it. It's the phosphate ions. So if we now go to the reactive oxygen species and see if they are involved in the fatigue in some ways too. And the production of these free radicals increases during fatiguing exercise. The likely source is supposed to be mitochondria, and you can read in textbooks and so on that if you use your mitochondria, 1 to 2% of the oxygen will become free radicals. And if you start to think about that truth, so to speak, then to run a marathon, for instance, would be absolutely lethal. You will have buckets of free radicals that your muscle and body can never take care of. So this is clearly not true, and it's much, much less than that. And I guess it's also heavily controlled. So now comes some hardcore free radical biology to show the players that we do have here. So the parent, the first one, is superoxide. And that can come from mitochondria, which are supposed to be the major source. But we also have the NADPH oxidase that's become very popular now because people know how to measure it. So some claim that this is really the key player to produce the superoxide. And the superoxide is a dangerous fellow, so you should try to stay away from that one, most likely. And the most effective way to get rid of that is SOD, superoxide dismutase. that turns this into hydrogen peroxide. If you're lucky, you have enough catalase, and then it will become water and oxygen. But you don't have enough catalase, so this system is rather slow. Another good way is to use glutathione, and then you also get water. But this is a limited source, so you don't. That can also be exceeded if you produce too much of it. If you're over here and unlucky, especially if you have iron around, you can get hydroxyl anions, and they are really reactive. They will react with anything that is in the neighborhood. And I think when they occur, there is problem. They're going to do something that you don't want to happen. Another thing is that you produce another radical also in muscle, nitric oxide, NO. And if you have a lot of this and NO, you get ONO. And you can hear from the name that that's also someone that is likely to do something. Peroxynitrite is the official name of it, and it's also very reactive. So ROS is supposed and has to increase during fatiguing stimulation, but when you try to measure it, it's very, very difficult. So everyone says that it happens, very few can measure that it actually did happen. We've tried with various fluorescent indicators, and many times it looks like this. So here we have mitosox red. That's a fluorescent indicator that measures the free radicals in the mitochondria. So here you have two muscle fibers. They are fatigued, and then we hope for more red signal here, which we can't, well, maybe if you look carefully, it's up by 2% or something like that here. We put them in hydrogen peroxide just to see that if the system is working, and then they really get red. So you have an indicator that does see something, but it's very, very difficult to measure an increase during fatiguing stimulation. But does that mean that it doesn't happen and everything is a big mistake, so to speak? Well, we tried then experiments with slow twitch, fatigue-resistant muscle fibers, so that they really use their mitochondria a lot, and they should really produce lots of free radicals to them. And then we also learned that the production of these free radicals would be worse if you do experiments at high temperature. So we take a Soleus fiber, we fatigue it with 600 milliseconds maximum contractions every 2 seconds. So this is pretty tough. And we do that 100 contractions, 37 degrees, where it should have a lots of ROS being produced, and not much is happening. Increase to 43 degrees, where basically skeletal muscles are supposed to be dead, but they aren't if you do it this way. And again, not much is happening, so they can survive this, and from there it looks like free radicals and high temperature and so on doesn't do much. We actually went up to 47 degrees and stimulated muscles, and they were perfectly happy there too. So apparently at least mouse muscle fibers can live at very high temperatures, because I think the wisdom is that the muscles will coagulate or something like that at 43 degrees. These guys didn't. So then we did something even worse. At 43 degrees, we also put on 10 micromolar of peroxide of free radicals. And this is sort of 1,000 times more than a muscle fiber will ever see in vivo. And did fatigue? Nothing happens. So it looks like during fatigue and contraction, this ROS is completely unimportant. But when we stopped, then there was problems. So they went into contracture after 15 minutes or something like that. So they were perfectly happy as long as they continued to be stimulated, but they sat down and rest, so to speak. Then the problems occurred. A bit similar to what you experience at the end of a marathon race, at least with the guys that aren't really the professionals. So people manage to run the last few hundred meters, perhaps not fast, but they're still running. And then they stop, and then they try to walk sort of 3-4 minutes later, and then it looks quite fun, I would say. The Stockholm Marathon, for instance, then you have to walk down a rather long stairs after the finish line, and to see people that now have been running for 42 kilometers at some kind of decent speed, trying to walk down the stairs. It's quite fun. If you should watch the Stockholm Marathon somewhere, it should be that stairs, because people try all sorts of crawling backwards. And so something happens when you stop. And maybe that's when the free radicals come into play. So then focus on the recovery related to them instead. And recovery after fatigue is often very slow, especially at low stimulation frequencies. And this was originally called low frequency fatigue, which if you read about fatigue and recovery and so on is a frequently used term. Unfortunately, that's used for almost anything now, so no one knows what it means anymore, and we try to change it to more complicated ones that shouldn't be misused. Prolonged low-frequency force depression, because I think that's really what it is. But it's more difficult to say, so PLFFD instead. And what's the underlying mechanism there? And if you translate this to something that you can understand, this is sort of being out of shape. Everything feels awkward. You have heavy legs, and whatever you do, you need a lot of mental input to do something. You can still perform, but it's much, much more demanding on the nervous system. You have to increase the stimulation frequency to get the movements going. So the fact that you have more problems at low frequencies than at high frequencies is simply because of this force-calcium relationship. So there are two ways that you can have this, basically, and that is that you have reduced calcium release, the red part here. So if you decrease calcium up at high stimulation, close to maximum force, not much is happening because you're on flat part of the curve. If you instead decrease calcium here, and here is where the nervous system normally works, then force declines very rapidly, and you have to increase the stimulation frequency from the nerves to get back up there, and that's sort of tiresome. The other one is calcium sensitivity. Doesn't matter up here because you're on the flat part of the curve, has an enormous impact on the steep part. So you have, again, if this is happening, then you have to increase the frequency to come up to there again. If we look at mouse fibers, so this is before and after fatiguing contractions. Force is down at 30 Hertz a lot, and calcium is down. 50 Hertz, force is down a lot, and calcium is down. 100 Hertz, force isn't really down that much, but calcium is down. So in mouse fibers, we have this PLFFD because of reduced calcium release, which has an enormous impact on the steep part of the curve, close to no impact at all on the flat part of the curve. So it's nothing mysterious that it happens at the low frequencies. It's just because of the shape of the relation between activation and force production. If we do the same experiment in the rat, so this is the force after compared to before. It's down to 30% 1 hour after fatigue at 20 Hertz and so on. And if you come up here, it's not much reduced. So it's the same PLFFD here. But in this case, there is no problem with calcium. So calcium is normal here. So in the rat, it seems to be calcium sensitivity instead. And in the mouse, it's calcium. So you can have two things, two completely different mechanisms with the same endpoint. And what we know is that the calcium sensitivity is very sensitive to hydrogen peroxide. And hydrogen peroxide versus superoxide depends very much on superoxide dismutase, the enzyme that translates superoxide into hydrogen peroxide. So the rat have more of it, meaning that in the rat, if you have superoxide, it will immediately be hydrogen peroxide. And in the mouse, it will stay as superoxide. And when measuring the superoxide during fatigue, it actually increases in the rat, but not in the mouse. So the conclusion from this is that During recovery, free radicals seem to be important. If you have an increase in superoxide, it seems like you decrease the calcium activation pathway there. And that's the mouse one. If you have an increased hydrogen peroxide instead, you get another problem. Then you have decreased myofibrillary calcium sensitivity. And that's the rat one. And where the human one is, I don't know. But what I do know is that this PLFFD is a classical phenomenon also in after severe exercise in humans. And I think it could be one of the most important factors behind overtraining and so on, that something happens here. And then if it happens on the calcium, on the mouse version, on this version, I don't know. But I'm pretty sure I'll come back to that the mouse version of it is one of the important players. So does this mean that the reactive oxygen are always the bad guy? Definitely not. And this is just one paper showing that it's not. So in this study, they gave the subjects high concentrations of antioxidants, and then they did endurance training, and absolutely nothing happened. Meaning you can get no effect of endurance training if you eat a lot of antioxidants. So these ROS are clearly one of the most important perhaps, signaling molecules to tell the muscles that, okay, we have a problem here, we have to adapt. We have to get more mitochondria, better defense against this and that. Luckily, with all these pills that you're recommended in the ads and so on to eat to increase your antioxidant defense and so on, they contain so little so it doesn't matter. So basically you're just wasting your money by buying it. But I mean, the advertising is really stupid, like I have a really active lifestyle so I have to use this, whatever. Why on earth would an active lifestyle all of a sudden be some kind of disease where you have to eat certain antioxidants to survive? I mean, it's so half criminal, I would say, but there is a new law or something coming that you can't claim that something is good for you if it's not proven to be good for you. And the antioxidants that are sold are clearly one example there, where if you have some kind of disease it may be good, but I don't know of any sort of hard evidence that it does anything good there either. If you're a normal person with an active lifestyle, I would stick with food. So, related to this, I am allowed to keep on for a few more minutes, I hope. Yeah. So, coming to training here, we asked what's really the mechanism behind that endurance training leads to increased fatigue resistance, based on this kind of ROS thinking and so on. And we used flexor digitorum brevis, our favorite muscles, of cold acclimated mice. And that seems a bit upside down. Instead of doing endurance training, we used cold acclimated mice. The idea behind this was first that we had them, so we could study it. The other one was that they get stressed too by living in a cold environment. But so they get the kind of stress you also get with endurance training. But with endurance training you get all the mechanical input. There are all sorts of things happening at the same time. So it's rather unclean, kind of, if you want to look at the mechanism. So therefore we did this as sort of a substitute. This is sort of endurance training without endurance training. And just the stress induced by this, see if that does something. And the hypothesis is that that the stress by cold exposure should be something similar to endurance training. And that will probably by reactive oxygen acting on the ryanodine receptor complex. That is the calcium releasing channel inside the muscle cells. That something will happen with that, which will increase the basal calcium. So calcium will be high in muscle cells all the time. Not so high so that you get a contracture, but higher than normal. That is known, that if you have a higher calcium, you trigger mitochondrial biogenesis. So you get more mitochondria if you increase the calcium. That's been done in various sort of non-physiological condition, but this might be then a physiological condition where you didn't put in a drug or anything like that. This is something that the cell does on its own, or the cell do on their own. And this will then lead to increased fatigue resistance. So if we look at the ryanodine receptor, so the calcium, when you activate a muscle cell, comes out of the sarcoplasmic reticulum through this very complicated channel complex. The basic there is the ryanodine receptor, but then you have all sorts of associated proteins bound to it. And this one can be phosphorylated and nitrosylated and God knows what. Normally, action potentials can activate this guy, the DHPR, which is the voltage sensor, that mechanically pushes on this one. This one opens and calcium comes out. But there are many situations where this one becomes sort of leaky and calcium comes out also when it's not pushed on. And we think that that's something that could happen with endurance training. And focusing a lot on this friend here, FKBP, which is a protein sitting here and sort of stabilizing the thing. And that may come loose. And then you have a more flexible channel that may open when it shouldn't open. So what did we see in our toe fibers of cold acclimated mice? So here we have then the wild type. And what we do here is that we take out the ryanodine receptor and then measure how much FKBP sits on it. And it's difficult to see, I believe, but here at 4 degrees, there is less FKBP sitting on it. And also in the UCP1 knockout, which you can forget about right now, these are mice that can't activate their brown fat. So when you put a mouse at 4 degrees, it shivers. So it uses skeletal muscle to generate heat, and after a while it stops shivering because then the brown fat takes over and generates the heat. Whereas the UCP knockouts, they can't do that, so they continue to shiver. But they don't shiver with the toe muscles, so the toe muscles don't care about the shivering business. So at any rate, so you have less FKBP on the cold acclimated ones. So that should then make the channels more leaky. And what we saw is then that the basal calcium went up. So they have a higher calcium. And if you have a higher calcium, you should get more mitochondria. And when we looked at that, PGC1 alpha, which is sort of the main trigger of making new mitochondria, is increased in the cold acclimated. Citrate synthase activity, a measure on how much mitochondria we have, is increased. No change in fiber type, which is also something that basically doesn't happen with training. You can change between type 2A and 2X, but otherwise you have the fiber types you have. And lots of literature says that fiber type switching from this and that occurs, but it occurs, in my mind, in very, very unphysiological situations when you cut the nerve and things like that. With normal training, they don't change. It's that the fibers you have become better on what you train and not anything else. AMPK, which is the metabolic sensor, nothing happened, so it's not because you change the metabolism that you get more mitochondria. And control mouse, the normal kind of fatigue pattern, cold acclimated one, much more fatigue resistant. So you can, by fiddling with the ryanodine receptor, have more leaky calcium channels. By cold exposure, you get then increased basal SR calcium release, increased calcium, more mitochondria, and increased fatigue resistance. And we believe that this could also be a sort of an important mechanism behind the endurance training and the triggering of endurance training. And also this thing, if you have an increased SR calcium release, an increased calcium, you need to pump this back into the sarcoplasmic reticulum. And when you pump something back, you get heat. And I think that's very familiar to anyone that's done some kind of endurance exercise, that you feel hot afterwards. So something has to generate the heat. And this, to me, is a very likely source. You have some leak there, you have to pump it back, And then also when you train hard and overtraining and so on, you can have an increased heart rate at rest. Again, if you have leaking SR calcium channels, you have to pump it back. That will cost energy, which means that you have to use your mitochondria more, which means that you have to pump a bit more blood too. So that could be sort of an effect that you can see, that when you have done something to your ryanodine receptor, you can feel it as an increased temperature. But does it happen with normal training too? We've just started, so this is really new data from us. And so we trained mice. They had access to running wheels for 6 weeks, looked at the ryanodine receptor, and FKBP disappeared from it in the trained animals. And they also got better mitochondrial, increased mitochondrial capacity. We tried it in normal mice and mice with tumors, tried to cure the tumors, didn't work, but at least they got more fatigue resistant. So this happens also with training. But what about overtraining? To have leaky channels might not be a very good idea. And then comes the shivering part in. So if you take the soleus muscle, which participates in the shivering, then it's exposed to the cold, plus it's shivering all the time. And in them you have a massive decrease of the FKBP bound to the ryanodine receptor. So it's down an awful lot here. So now you really have a leaky channel, and if you look at the function there, calcium is down, force is down, and these guys aren't happy, so they have very weak muscles. So now you sort of turn something good into something bad. So if you really overdo it with endurance training based on this kind of scenario, then so when you have cold exposure and shivering, you have a massive calcium leak, decreased calcium, muscle weakness, and this could be sort of the overtraining aspect of it. So everything in biology more or less has a curve like this. Little is good for you, a lot, then you come down on the other side, then it's bad for you. And the very last thing then, which comes out of our research the last few weeks more or less, is that nitrate has become a hot substance here. So nitrate feeding, and the nitrate feeding in this case is like eating a lot of green vegetables. So eat a portion of spinach or lettuce or so a day. What we've seen that in fast muscles over here, you have an increase, so the open symbols is with nitrate feeding for a week in mice. So you have a lot more force at low stimulation frequencies after eating this, after feeding them with nitrate. You don't see it in a soleus, a slow twitch fiber, but in a fast twitch muscle this happens. So this is the opposite to this PLFFD. So instead of having lower forces here, you have bigger. So this would then be that you really feel that you are in top shape. So now nothing can stop you. The mechanism is that calcium is higher. So at 30 Hertz here, you release more calcium. So the calcium machinery has improved. The nervous system then don't have to work as hard to get the same kind of force. And when putting these guys, the nitrate-fed mice on the running wheels, they run like mad. So they run about twice as long as the other ones. So they run something like 22 kilometers each night. Normal mice like to run around 11 or so. So this is something that the muscles clearly improve. If this is true in humans too, don't know. I mean, nitrate has become very, very hot recently. It's supposed to reduce your blood pressure a bit, maybe does something to the mitochondria, but maybe it also does something to muscle function more directly. Now I have gone over time severely, but this is the end of it. So thank you for listening.
Mittuniversitetet. Upptäck dina möjligheter.